Okay, sound working all right? Yeah? Yes, okay, thank you. All right, thank you so much for the warm welcome. I'm really excited to be here. I've actually never been to UCLA before, so this is also my first visit here. Hopefully I'll have some time to tour the campus a little bit before we go. So he mentioned my title. I'm going to talk today about stress, resilience, and embodiment. I'm going to broaden it a little bit more than this and not just talk about Hispanic immigrants, but talk about another project with Romanian orphans as well that some of you might have seen, a paper I published a few years ago, and I thought it'd be interesting to show you that as well. And I want to start by just outlining my perspective and my framework for the research that I do. So I'm interested in biological embedding of stress from a broad perspective. And what I mean by that is how social environment, social inequalities, things like discrimination and poverty, all different kinds of stress can get embedded under the skin, as the saying goes, to affect your biology and affect your health throughout your life course. So all different stages of life. This is an idea that's grown out of social epidemiology and anthropology, psychology. It cuts across many different fields. There are many subtle different ways of approaching this topic. I've been trained as a genetic anthropologist, so I'm taking my own unique genetic anthropology approach to this research, and my focus is on epigenetics specifically. So I'm interested in epigenetics as one pathway, one mechanism through which the social environment can become embodied into our bodies. And one more definition of embodiment here coming from Nancy Krieger, literally how we incorporate biologically the material and social world in which we live from conception to death. So I like this definition of embodiment. It's taking the material and social world, the political, social, economic context in which individual bodies are placed, and showing how these factors that our lives are nested within can affect our health through different ways. And so I'm interested in the epigenetic pathway in tandem with the genetic pathway interacting at the level of gene expression to affect the phenotype of the offspring throughout the life course. And I'm interested in all different aspects of the social environment. So I show here an image of toxic pollutants, but I'm interested more in the toxic stress side of the environment, and I think of all of that as interrelated, and people's economic, social status influences these exposures in their environment, and all of that affects these pathways towards health. Okay, so I'm sure most of you have heard of epigenetics by now, and you're familiar with this basic definition, but just in case, I thought I'd go through briefly. Epigenetics are chemical modifications to the DNA that can potentially alter the way the genes get expressed. And when people talk about epigenetics today, they're mainly focusing on three types of mechanisms. DNA methylation, which is the most studied, most stable type of epigenetics that I study, and that's here little methyl groups, CH3 chemical groups attached to the cytosines in your DNA that precede guanines in the genome. So C's that precede G's could potentially have a methyl group attached to them, and these methyl groups could alter the availability of the DNA to be expressed into a protein. Also, histone modifications are the second main mechanism people talk about, and those are chemical changes to the amino acid tails that come off of the histones. So histones are the little proteins that your DNA is wrapped around, and then they have these little tails coming off of them, and different chemicals like methyl groups, amino groups, or acetyl groups can attach to those amino acid chains and change the way the DNA is folded up and make it actually more unfolded and more available to be expressed. So the third one I didn't picture here is microRNAs, which can also bind to DNA or to RNA and alter the availability of RNA to be translated into a protein. So the two most people study are methylation and histone modifications, but like I mentioned, the methylation is more stable, more commonly studied, easier to study. So it's more done on a population level than histone modifications are at this point. And so my focus is on DNA methylation, and I just wanted to show you briefly, when we think about methylation, we're interested in regions that might have functional relevance for the expression of a gene, and often we start with looking in the promoter region, which is the region just upstream of the transcription start site of a gene. And we look for CPG islands, which are clusters of CG sites that tend to occur together in a densely populated area. And if they're methylated, like in this picture, then they could potentially block expression, block a transcription factor binding protein from coming in and binding and block the transcription of that gene. If they're not heavily methylated, then it might be more available to be expressed. And I always say this with a grain of salt, because this is a really oversimplified schematic of what's happening. I've actually never seen this one-to-one correlation of methylation with expression. It's way more complicated than that. Other epigenetic factors, genetic factors interact to affect what gets expressed higher or less. And just looking at methylation doesn't always predict expression very well, just as a cautionary note. I also forgot to mention, I'd be happy to take any clarifying questions throughout the talk if you'd like to ask. We'll hold the more in-depth questions for the end. Okay, so I'm going to tell you about some epigenetic data today, but before we get into the data, I wanted to just show you one perspective. I'm interested in epigenetics in part because of this idea of developmental plasticity, which is potentially could be considered an added layer of complexity to genetic diversity and the evolutionary complexity of humans and other animals. So by developmental plasticity, I mean the process by which developing fetuses and children adapt to their environment within their lifetime. And epigenetics may play a role at this level of evolution by allowing a fetus or a young child to adapt to its local environment and fine-tune different regulatory set points that might be established genetically. And what's nice about it is that it's at this intermediate level of timescales. So this is a really nice figure from Zane Thayer and Chris Kuzawa's paper about timescales of adaptation, and it shows how developmental plasticity allows for this fine-tuning of regulation, maybe regulation of immune function or neural function or stress response systems that help them adapt to a particular environment that happens faster than typical evolution, where allele frequencies change rather slowly to adapt over millennia to an environment, but slower than homeostasis adaptation, something like climbing a mountain, having low oxygen. Your heart rate might increase within a very short period of time to help circulate your blood flow better and reach more oxygen to your brain. So that's the quickest level. Genetic allele frequency changes are the slowest level, and epigenetics might play a role in this developmental plasticity intermediate level. Okay. So one last thing I wanted to say about the developmental plasticity perspective is that there's a chance that this may be adaptive in a beneficial way in some cases. I actually think in most of the cases it's just harm. Toxic exposures like pollutants or stress are harmful. They overwhelm the system and lead to more vulnerability, risk for disease. But in some cases there could be an adaptive benefit to developmental plasticity, such as a case here where you're regulating set points of stress response in a mouse, say. If a mouse was exposed to toxic stress in pregnancy or in childhood and it becomes more cautious, is more sensitive to threat, it might be safer for that mouse to stay at the perimeter of an open field, as a psychologist would test a mouse's exploratory capacity. It might be more cautious, it might stick to the perimeter, and that could be an adaptation to protect it from predators. So it could be beneficial in some cases, but I think if you think in terms of humans today, the environments that we're in, it's probably mostly not beneficial to be more sensitive to threat and more be more vigilant. Could be beneficial, but I think most of the time it probably makes them more vulnerable to mental health disorders down the line. But there is evidence that children exposed to abuse might be more hormonally sensitized. Their amygdala is more reactive to threat. They recognize anger more quickly when they see angry faces. So in some period in our evolutionary past, that could have been an adaptive change to protect them from predators or something like that, or just from threat. Okay, so today I'm going to tell you about two projects involving epigenetics that I've been a part of. The first one is the Romanian Orphan Study, which is the Bucharest Early Intervention Project, and I was really fortunate to get involved in this project when I was a postdoctoral fellow at Harvard. It's a collaboration between Harvard, Tulane, and Maryland. It's been going on for 18 years now, so it's a really rich, exciting, unique opportunity to study early childhood environments in a really controlled way where it's a randomized controlled trial of institutional care that's really unique. So I'll tell you about a little bit of epigenetic data we've published on that study, and then I want to talk briefly about my own project that I started a few years ago, studying embodiment of stress in children of Hispanic immigrants. So before we get into the details, I thought it would be nice to lay out some of the major challenges in this field of research for those of you interested in studying it or just reading about it, and help you understand the framing of the choices we had to make in designing these studies. So epigenetics is really different than genetics because if you look across all the cells in your body, everyone has the exact same genome. Any cell you look at, you have the same information, the A's, the C's, the G's, and the T's in the same order. But epigenetics is different because every tissue type and cell type is potentially different. In fact, what drives cells to differentiate from each other are the epigenetic signals that turn on or off genes in different ways in different cells to create different types of cells. So every cell type is potentially different, and that's something you have to worry about when you're collecting samples to study epigenetics, because the choice of tissue could really change what you're seeing, the pattern of epigenetics that you see. So when you're thinking about what tissue to use, most people study the peripheral tissues, which are blood, buccal cells from a cheek swab, saliva from a spitting into a tube. They're the ones that are easy to get. But they might not be the ideal tissue for the disease of interest, the exposure of interest affecting those, where genes are actually being expressed in those tissues or not. So I want to talk about that briefly. Also, you need to worry that you're getting a mix of different cell types in every kind of sample that you take, particularly if you're studying blood, because you've got all these different kinds of white blood cells mixed together in a whole blood sample. And unless you sort them by cell type, which is really expensive, you've got to worry about the fact they're all mixed up together and you're getting an average of the methylation patterns across these different cell types, which could be very different directions, and you're just swamping out any signal you might have found in one particular cell type. So that's why I shy away from blood. There are, well, that's one reason. There are bioinformatic techniques to adjust after the fact for different proportions of different cell types in your blood. Based on the methylation patterns you see in the blood, you can infer the proportions of cell types you had and then adjust for that and look for a signal that goes beyond the cell type proportions. But if you're working with buccal swabs with epithelial cells, you don't have to worry about that so much because you're only getting one more homogeneous cell type. You also have to worry about the timing. Unlike genetic studies, with epigenetic studies, the timing could matter. It might be that you sampled a child too early before a sensitive window in which their methylation pattern could change, or too late where it didn't last that long. So we are still trying to figure out when are the important time points for when cells are more vulnerable to epigenetic change, what exposures matter, and what time periods. So just something we're still trying to figure out. And then your question might be about mood regulation or psychiatric disorders or PTSD, and you're sampling blood or saliva, and the question comes, is that really relevant to the disease of interest? It's convenient, but is it really important? If you're thinking of looking in blood as for a biomarker of an exposure, it might not matter, but then again, that tissue might not be affected, so you might be missing it. Okay, so just on this point about the stem cells differentiate into different kinds of cells, you have to worry about which cell type you're capturing. I am going to tell you about my studies. We looked in saliva and buccal cells. And one other reason I think that that's a good sample to look at if you're limited, if you can't get brain tissue, which would be the ideal, is because they come from the same ectoderm layer of origin. The The epithelial cells and the neural cells actually are both part of the ectoderm, whereas blood cells are part of the mesoderm. So you're getting something that has the same embryonic origin, and there have been studies that show saliva and buccal might be better surrogate tissues for epigenetic patterns in the brain than blood samples might be. And one more point on that. There's been a couple of studies looking genome-wide at epigenetic patterns across cell types, and they find that blood is pretty unique relative to the rest of the body's cell types. In this clustering of all the different tissues you could possibly test, and you can't read them, but the point here is that the blood is clustering separately than brain cells and buccal cells, or epithelial cells in the buccal sample. So for that reason, I think buccal and saliva are more interesting for psychiatric related diseases, if you're interested in those, or stressors that might affect mood regulation, that kind of question. If you're interested in circulatory disease or immune function, you still might be interested in blood, but if you're trying to get a proxy for brain, I would stay away from blood. Okay, the other thing I just wanted to point out when we're setting up these studies is that we don't know where to look in the genome. We're figuring out, this is a similar problem with genetic studies, same problem with epigenetic studies, what genes matter, what parts of the genes matter, because here we're worried about regulation of expression, not just what genotype you have. So you want to pick a gene that's relevant to your research question if you're picking a candidate gene, and that can be hard to do if you don't know what genes are relevant. So if you don't know, you want to look genome-wide and try to figure that out. But even when you're looking genome-wide, you don't know what parts of the gene to look at. So you want to look at least regions that are relevant to function, things that might be in a transcription factor binding site or a promoter or an enhancer region of a gene. And then you got to balance feasibility and cost, because these assays, if you want to look genome-wide, the easiest and cheapest way to do it is with an Illumina microarray that captures 450,000 CG sites in the genome. Now they're up to 800,000 sites in the genome, but they're expensive. They cost around $400 a sample. So if you have a big population level study, you can't really afford to do that on a huge sample unless you have lots and lots of grant money. So you got to balance that. And even if you do want to do that genome-wide approach, it's only capturing less than 1% of the CG sites in the genome, because our genome is really big, has about 4 billion sites in it. So even there, you're missing a lot of relevant sites that we might be wanting to capture. Okay, so in my studies, I focused on genes related to the stress response system, the hypothalamic pituitary adrenal axis, which is the primary stress response system in our bodies. And I've taken a candidate gene approach to that, looking for genes that are candidates to be relevant to this HPA axis. And just to outline, in case you haven't seen it in detail before, the HPA axis is just the system starting with the hypothalamus, where stress signals come into the brain, sends a cascade of hormones through the body to the pituitary and adrenal glands, ending with the release of cortisol, which is our stress hormone. I'm going to talk about cortisol today. Cortisol has lots of downstream effects on immune function, on glucose metabolism, all kinds of things. And it's important for this regulatory feedback loop. It sends a message back to the hypothalamus to downregulate the expression of more cortisol so that you can recover from a stressor relatively quickly by regulating this feedback. So the genes I'm interested in are part of this feedback regulation, and if they get more or less methylated, it could disrupt this feedback regulation and cause it to be more difficult to recover from a stressor. Okay, so most of you have probably heard about the rat licking and grooming studies, which made social behavioral epigenetics famous in 2004. Michael Meany and colleagues published the foundational study in this area about how the rats lick and groom their pups at different rates. And the mothers who lick and groom more frequently, you can think of it almost as a model of maternal care. The more attentive maternal carrying mothers lick and groom more frequently end up with less methylation at this particular glucocorticoid receptor called NR3C1 that's part of that HPA axis system. In the hippocampus specifically, you see less methylation and more expression and lower anxiety in that rat pup as it grows up, and then a recapitulation of that behavior, they lick and groom their pups more across the next generation. So it's a behavioral transmission through the epigenetic path of changing methylation at this one region of the brain. And those that lick and groom less actually have more methylation, less expression, higher anxiety, and less licking and grooming in the next generation. So most people have heard about this. It really set off an explosion of research trying to replicate this in humans and other animals, seeing what other genes might be similar to this. And I just wanted to take a moment to talk about how well has this study been replicated. It's a question I've been asking myself for a while now, and I've heard of a lot of people having trouble replicating this, myself included. And I would say yes and no, this has been replicated well, depending on how you define replicated too. So there's a great review article by Michael Meany and Turecki looking at all the studies that they think were important for studying methylation at this particular gene. And this top panel showing you the animal models, and this is showing you human models trying to replicate the finding. And they found that it's been pretty well replicated at the same exact exon in the same exact gene in animal models. 7 out of 10, I think it was, yeah. And 9 out of 10 human studies have shown increased methylation at this particular gene related to early life adversity in children or in animal models. But in humans, there was one study that found no change, one study that found increased and decreased at a different site within that same exon. So there's a little bit of diversity, but most of the studies, most of the research is showing increased methylation at this particular region of this particular gene. And these are human studies across different peripheral tissues, blood, saliva, not brain, brains and blood and other things in the animal models. So it's been kind of pretty well replicated. But I would argue that the magnitude of the effects in humans is so much smaller than it was in the hippocampus of the rat brains that we still don't know for sure how functionally relevant these changes are in humans, at least in peripheral tissues in humans. Because the size of the change is so small compared to what they saw in the rat brain, and we don't know how biologically significant those changes are actually going to turn out to be. And it's really difficult to test those in humans. So I think it's yet to be determined that any peripheral tissue is going to show that effect. And maybe it is changing the brain. And there's one study of human brains after death that shows a change. But in blood and saliva and buccal swabs, it's very minimal, the amount of change that you see. So I also just wanted to talk about this just because I find this to be fascinating. It just came out a couple weeks ago. The biggest genome-wide study to be done on early life adversity. I've discussed this with my lab students and with everyone I come across now because I think it's going to be a really important study that showed negative results genome-wide with the most comprehensive measures of adversity that they could get on the biggest sample I've ever seen looking genome-wide. So this was a study by Sarah Martzi. The senior author is Avshalom Caspi. They did a 2,200 person twin study with extremely comprehensive measures of childhood and adolescent adversity. They call it victimization. They accumulate across all different types of adversity, or victimization, things like sexual, physical, emotional abuse, neglect, child maltreatment, victims of crime, witnesses of crime, bullying, any kind of victimization you could think of, put together in childhood, separately in adolescence, cumulatively. They measure it prospectively, which is pretty much the gold standard, and retrospectively, and they compare the findings. And here is the Manhattan plot of what they found. Actually, this red line is the level of genome-wide significance for anything that's important after correcting for all the multiple testing that they did across 450,000 sites in the genome. There were only 2 sites that crossed that threshold, and after they adjusted for smoking I think in the parents, maybe in the children too, they found none of the sites passed that genome-wide significance threshold, and smoking explained those 2 sites because smoking was correlated with victimization experiences, so it was confounding that analysis. So their conclusion is that with the most comprehensive study yet done, with the largest sample size, this costs over $500,000, probably close to $1 million to get that many samples with that much data. They found nothing, and they argue that maybe we're not going to be able to replicate the findings in blood. They looked in blood that we see in these animal models in the brain, and either we're just not doing it right, or there really are no effects big enough to detect in a genome-wide scan. So just to note, they also looked within that same gene in the glucocorticoid receptor that was found in the rat pups that have been licked and groomed. And they looked at every single site in the genome-wide scan within that gene, and they didn't see anything that passed gene-wide significance for the number of sites within that gene for any type of adolescent, childhood, prospective, cumulative, retrospective victimization. So they don't see it within that gene, and they don't see it genome-wide. You could argue with the way they multiple tested, corrected for the whole gene region. Maybe their hypothesis should have focused specifically on the site that was in the mouse model, and that would have shown a different result. But in general, their conclusion is that there isn't anything significantly associated with victimization in childhood in blood. So it's possible all of the studies have been small up to now, and the findings that are exciting in this field might be false positives. I don't think they all are. I think we need more focus on hypothesis-driven candidate genes rather than just genome-wide explorations, where it's really hard to pass that genome-wide significance threshold when you're doing 800,000 tests. And if we start with a hypothesis, you can zoom in on the region that might be relevant to your exposure or your disease. Maybe the effects are going to be really small in these peripheral tissues and really nuanced in particular environments with particular interactions between genotype and environment and epigenetic, and you need better measures of the environment to really detect them, and you need bigger samples to test for interactions. That's where anthropology can come in with more deep ethnography of the cultural meaning of the environment to the people. And maybe we just need more rigorous, detailed measures of those environments. So the tests used in this study were standard psychological questionnaires. Maybe they weren't, it was a British study, I forgot to mention, of British kids, and I believe only British kids. So it would be, there's not much cultural diversity there, but it could be that there's differences in perceptions of stress across children that could affect the way that they report these exposures, and that's something that anthropology could help contribute to getting more rigorous, detailed measurements of the environment. Or maybe the problem was just blood. Maybe blood wasn't that good of a tissue to look, and maybe we should shift our focus to other tissues. So all of this to say is I'm starting to think about these things more deeply now, and whether or not we should keep doing the same kinds of epigenome-wide studies and the same kinds of easily accessible tissues. Maybe they're not leading us anywhere that we had hoped they would be. Maybe the field's been a little bit overhyped. I'm still doing it, and I still think we need to pursue it to figure out if it really does matter or if we should change direction. But I just wanted to let you guys know that I'm cautious about the results, and I would interpret all of those findings you read with more caution, because small sample sizes could affect these findings. The choice of tissue affects the findings. The rigor in the measure of the environment can affect them. All of these things are important. So with all of that, yes?
Sorry, can I ask a quick
sure.
clarifying question? Was the sample trends diverse in terms of the amount of diversity? I mean, like, was there enough diversity within the sample in terms of their exposure to childhood diversity, or were they?
Yeah, I think they argue they had really good diversity. They had diversity, they argue, in social economic status, and I think they had good diversity in victimization. And I actually talked to Caspi about it through email and asked him a few questions about the study, and he said he really believes their measures of victimization were strong because they're correlated with mental health outcomes down the line. So he didn't think the problem was with the measurements. He thinks it's more that behavioral things like smoking and diet, exercise might be mediators of these epigenetic changes, and we might need to focus our attention more on those rather than a separate biological effect independent of behavior. Yeah. Which I think is interesting, and I agree, smoking seems to matter a lot. I'm studying those things as well. Okay. Okay, so in my two studies, I took a candidate gene approach to start with. I'm also moving towards a genome-wide approach, and I think we need to go back and forth. But I have candidate gene results to show you in this study. And I also looked in saliva and buccal swabs rather than blood. It's also hard to get blood from children, so it made sense. I'm going to start with this one. I was going to say, I think that was it. Yeah. Okay, I'm going to start with the Bucharest study. So this one's been published, and I think I forwarded the article on to Avery so he could distribute it if you guys want to read the article as well. This is the unique project that took place in Romania starting around the year 2000, and it was that collaboration between Harvard, Tulane, and Maryland, and it's the first, maybe only, randomized controlled trial in the world of the effects of institutionalization and foster care on children's development. And this study began in Romania in part because in the 1970s, the dictator established a regime that encouraged people to have a lot of children, in part because they wanted an independent workforce. And so they penalized people with taxes for having fewer children and rewarded them for the more children they had. And in the end, they had a really large population that not everyone could afford to feed and to raise, and they encouraged, they built a lot of orphanages to help raise these children and encourage people to use the orphanages if they needed to. And at the time the study began, it wasn't actually clear that orphanage care under age 2 was harmful, and they invited the researchers in to do this study to try to figure out that question, if it was harmful for the children's development. This was the design of the study. It was a randomized controlled trial in the sense that they collected samples from children living in orphanages and then randomized them out of the orphanages into a foster care setting, a foster care group that they developed in Romania. No foster care system existed at the time, so they developed a really nurturing foster care environment and then randomized the children into it and followed the care as usual group as well as the foster care group over time, in addition to a never institutionalized group from the community, and measured everything you could think to measure: brain function, structure, MRIs, blood or saliva samples, and growth, development, psychiatric tests, attachment, emotion regulation, everything you could imagine wanting to measure across toddlerhood up to 16 years. I'm sure they're still studying it beyond, I think they're 18 years old now. So it's a really unique design with a really rich sample. And anytime we talk about this, it's important to mention some of the ethical issues that get raised in a randomized trial of this type. And I just want to go through these quickly to address any questions you might have. So there's a lot of question about exploitation. People always ask, is this an exploitative study going into a setting of vulnerable children? And my take on that is that the researchers were invited into the setting. They didn't come in to do the research. They were invited in by the government to do the study. So I don't think that the motivation of this was exploitative. They had a non-interference policy, so if someone were to be adopted out of the orphanages or out of the foster care system, they did not interfere with any adoptions or any placements of the children. The risk-benefit ratio actually is in favor of benefits because there was no foster care system in place at the time. So anything they did was to help some of the children. Unfortunately, they couldn't help all of the children, but they helped a lot of the children. And then after the study ended, they didn't interfere with any of the placements of the children. The foster care system continued. I think the funding of the training of the parents ended, but the foster care system continued. Are there any questions about the ethical issues before we go on? Okay. I guess I addressed them. Okay, so I just wanted to highlight a couple of the findings before I get to the epigenetic results that were really interesting and fascinating of the changes they found in kids that were in the foster care versus the institutions. They found those that stayed in the care as usual group, the institutional group, had deficits in height and weight and cognition, social emotional behaviors, psychiatric disorders, differences in their brain structure. Actually, the amount of white matter in the brain differed. In the never institutionalized group, there was more white matter than in the care as usual institutional group and the foster care group. different electrical activity. There was even a study about telomeres by my collaborator Stacy Drury, who measured telomere length, the repetitive DNA at the ends of your chromosomes that shorten as you age. They actually found shorter telomeres with more institutional care, more time spent in the institutional care, particularly among boys in the study. A lot of these findings were reversible in the foster care setting when they were randomized out of the institution, so there's hope that an early intervention can actually benefit children in these situations. And at the end of a lot of these studies, the system in Romania actually changed to not allow children to enter institutions under age 2 once they realized how harmful that could be for children's health. So I think there was a really good benefit to this research. Okay, so I entered the study at the time we were working with the leftover DNA that had been extracted from saliva that had been used for looking at telomeres, and I wanted to look at a couple of candidate genes related to stress response in the children, and I focused on these two, the serotonin receptor and FKBP5. So FKBP5 is an important regulator of the glucocorticoid response system. Studies of FKBP5 with childhood maltreatment had actually found decreased methylation in children with child abuse and increased expression of the glucocorticoid receptor and a decrease in the efficiency of that negative feedback regulation of the HPA axis. Serotonin is an important neurotransmitter for mood regulation, and the serotonin transporter is important for removing circulating serotonin from the synapses between your neurons. And so other studies had found maternal depression during pregnancy actually could affect methylation of the serotonin transporter, increase expression, and decrease circulating serotonin in the body. So I focused on these two genes. I didn't focus on NR3C1 in part because the level of methylation in that glucocorticoid receptor is so low in human peripheral tissues, like less than 5%. I thought we'd be more likely to find something in these other candidate genes that are related to the same processes. And we did find a small but significant change in the children, or not a change, a small but significant association between the amount of time in their life spent in the institution and the methylation levels at a few different sites in these genes. So I'm showing here plots of the percent time spent in the institution correlating with methylation at one of the sites. We looked at 2 sites only in FKBP5, and one of them showed this relationship. We looked at a few sites in the serotonin receptor and saw 2 of them showed this relationship. So it's never all the sites, it's never a big effect, but there are some effects at some of the sites that are locus specific, timing specific. We looked at the percent of their life spent in institutional care at different time points in their life in relation to methylation measured at one time point at age 12 when the saliva samples were taken. So this is methylation at age 12 associated with percent of their life spent in institutional care at baseline, at 30 months, and at 30 months. And those were the significant results. So these plots are only showing the children that were ever institutionalized, excluding those that had never been institutionalized, because the effects were stronger within the ever institutionalized group than the whole group. But we saw the associations in the whole group as well. Okay, so to summarize these findings, the time spent in institutional care was negatively correlated with methylation in both genes, a few different sites at age 12. And this is consistent with altered cortisol reactivity that had been found in these children and increased risk of psychopathology that had been found in these children at age 12. There were different timings for the different genes, meaning how much of their life had been spent in institutional care measured at different points in their life mattered differently for methylation at age 12. So that might indicate that there are different sensitive periods that are gene specific that could differ between the genes that we're looking at. And I would argue we need a lot more research about causal pathways. We weren't really fully able to take advantage of that group structure of the data, the never institutionalized group, foster care group, and care as usual group, to get at this causal question, because not everyone spent the whole duration of the study within those group definitions. It was actually better to look at the percent of their life spent in care rather than the group designations because they sometimes left their group early or entered a group late. So we don't really have our evidence of causal mechanisms here. I would think we need more longitudinal and experimental studies to really establish that. But there is some suggestion that these methylation changes might play a role in this increased psychopathology that we're already seeing and might see more in the future. Okay, so I want to move on to talking about my second study. Unfortunately, I don't have epigenetic results to share with you because it's still ongoing, but I do have some interesting results about HairCortisol I wanted to present. So I guess I could pause here if there are questions about the first study to start. Yes?
What kinds of psychopathology were elevated? Was it more internalizing or externalizing or both?
You know, I'm not an expert on that area, but I think it was both. I think it was both, but don't quote me on that.
Thank you.
Yeah. Other questions? Yes.
The kind of material was preserved during how many years? The material that you used, the saliva?
Oh, the saliva was collected at age 12, and they were only a little bit older than that at the time that I measured it. So maybe a year or two. Yeah. It wasn't from birth. Yeah. Okay, we can come back to this at the end if other questions crop up. So I started this study in Nashville when I was a professor at Vanderbilt between 2012 and 2015. I got a grant to look at embodiment of stress among Hispanic immigrant mothers, among children of Hispanic immigrant mothers. And I spent a year doing a bit of focus groups, exploration, observation, trying to really fine-tune my measurements for this particular community. And one of the papers I shared with Avery right before the talk talks about the findings from the first phase of the study, which I'll share with you today. But I just wanted to highlight the fact that it's a multi-phase study and we're still in process. So the reason I was interested in Hispanic immigrants in particular is because they are the largest U.S. minority and growing. They're 17% of the U.S., projected to be 30% of the U.S., I believe, within 30 or 50 years. They're the lowest socioeconomic, they have a low socioeconomic status relative to other groups. 25% of Hispanics live below the poverty level, and a high proportion of them are uninsured, so they're at high risk for health disparities. 40% foreign-born Hispanics are uninsured. And a lot of people are interested in this Hispanic health paradox, where despite having a low social economic status, they actually have better health outcomes for some diseases among some groups than comparable groups of white Americans, such as birth weights. Their birth weights are better, less low birth weight than similar SES level white counterparts. So I don't really like this Hispanic health paradox idea because I don't think it's really true. It's not true across most health outcomes we measure, and it's not true, it doesn't last. This healthy advantage in birth weight doesn't really last throughout the lifespan. We know Mexican American children are actually 4 times more likely to be overweight or obese children than white children. So here's just showing you prevalence of obesity in Mexican American males and females, having the highest prevalence of obesity relative to non-Hispanic whites and blacks and other groups. So they're at higher risk of obesity and overweight, and so even if their birth weights start out healthier, they tend to get other metabolic problems later in life. So one question is this health disparity genetic? Is it coming from a genetic difference related to their ancestry? And I don't believe there's any evidence of genetic determinants for greater morbidity or mortality among Mexican Americans, but we do have a ton of evidence that their health is determined by social factors like income or duration of time lived in the US. So here's one example. You can see across all the different income categories, you see those who have lived in the US for less time actually have less risk of this Framingham risk score, which is a proxy for cardiovascular risk, cardiovascular disease. So those who have less time lived in the US are at less risk for cardiovascular disease. So duration of time lived in the US actually increases your risk of cardiovascular disease. And then across the income groups, those who have less income have higher risk than those who have more income. So if factors like duration lived in the US and income can influence their cardiovascular risk so much, clearly points to non-genetic factors for influencing this health disparity. So I came back to thinking about the stress response pathways and the HPA axis, and maybe this has a role to play in how their lived experience in the US could be changing their health outcomes. And I'm interested in looking at genes and epigenetic effects in these same stress response genes. Also interested in cortisol itself, levels of cortisol and how they might be affected by stress experiences. So I developed the study I call Chicos Children of Hispanic Immigrants Collaborating to Overcome Stress, because you always need a good acronym for your study. And the phase 1 was gathering information about stress experiences in a small sample, focus groups of Hispanic immigrant moms and kids. Phase 2 was to begin studying biological embedding of stress through these different epigenetic pathways, cortisol. I was interested in blood pressure as well as aldosterone and all different kinds of cardiometabolic biomarkers of health. And this is still in progress. This has also begun now. So since my study started, so much has changed in the political context, and I thought this was a great opportunity to go back to Nashville, see how these same children are doing after the presidential election, see how the new policies and rhetoric towards Hispanic immigrants has changed stress experiences, maybe altered epigenetic factors, cortisol. So I'm right now in the process of returning to Nashville. We collected our first 20 samples last month. The great news is that the families are all eager to participate, and everyone we've been able to reach, except one family, has been interested to participate again. We do really long 2-hour, sometimes 3-hour interviews with these families, with the moms, and like an hour with the kids. We collect height and weight and saliva and hair and buccal swabs and ask them lots and lots of questions. So it's great that they're interested and still participating. A lot of them are, of course, eager to help promote this research and this question to help their children and their communities. So you might be wondering, why is Nashville even an interesting place to study Hispanic immigrants? Of course, California is also an interesting place, but you might be surprised to learn that Nashville's becoming an immigrant hub, and a lot of places in the Southeast US are actually exploding in immigrant growth in recent years. So this is showing between 2000 and 2010, the frequency of Hispanics in Davidson County, where Nashville is located, has gone from 3 to 5% to 5 to 11% just in 10 years. And there's been 92% growth in that time period, the third biggest state to have growth in immigrants, and 145% increase in children living with immigrant parents in this region. And even more important for my interest in children and early life adversity, though only 10% of Nashville is Latino, 20% of the public schools are Latino, because the number of children is increasing and exploding. So the future of Nashville will have more and more Latinos in it. And so it's an interesting place, a new immigrant destination that will be interesting to one day compare to San Diego, say, which is more of a traditional immigrant destination that might have different context for how the immigrants are integrated into the community. Okay, so this is the huge schematic overview of my study design and what I wanted to look at when I started. So everything related to psychosocial stress related to immigrants, which includes racism, discrimination, social isolation, social status, exposure to violence, changes in parental marital status, any kinds of environmental contexts that could affect stress in immigrant families. I wanted to look at HPA axis dysregulation, the genes related to stress response, but I also wanted to look at other things related to cardiometabolic regulation, things like blood pressure and aldosterone, which is a hormone very similar to cortisol, but less well studied, that's related to cardiovascular health and stress, as well as blood pressure, BMI, waist circumference, and HairCortisol, which are the first results that I have to share, and I want to share those with you today. So I was interested in HairCortisol because it's a nice measure of chronic stress exposure over time. And I think all of these things together, whoops, all of these things together contribute to risk for cardiometabolic disease as these children grow up. So the children in the study were between the ages of 6 and 13 when I started. Now they're between the ages of 8 or 9 to 15 or so. And we can measure all of these things over time and see how they're changing, which is what I'd like to do next. So HairCortisol, I'm sure a lot of you have heard about it. It's really exploding in the areas of research that are taking advantage of HairCortisol to measure systemic levels of HPA activity, because we think that it's tracking with the cortisol levels in blood and in circulation. And what's nice about it is you don't have to worry about fluctuations throughout the day as you do when you're trying to capture the diurnal rhythm in saliva or urine. You can measure it at one time point and sort of get a log over the past month of the accumulation of cortisol in the body over time. Each centimeter of growth of your hair represents on average about a month of growth. So you're capturing about a month of cortisol exposure with each centimeter of growth. Typically people measure 3 to 6 centimeters to get 3 to 6 months of growth. Beyond that, it's not stable necessarily, and it doesn't last, so it could degrade over time. So 3 to 6 months is, I would say, the max. Most people think it's safe to measure cortisol. But it's a great way of getting a longitudinal assay of stress over time, or a change in cumulative stress over time. And I just wanted to mention one of the earliest studies of hair cortisol was actually done in these rodents called rock hyraxes. I just found this recently. They looked at social rank in hyraxes and found hair cortisol from hind leg hairs that they were able to get from captured hyraxes correlated with, the hair cortisol correlated with social rank. In the singing hyrax males, the ones who sing to find a mate, there was a correlation with cortisol and social rank. The more dominant ranked ones had higher levels, or lower levels of cortisol, higher social rank, higher levels of cortisol. So I guess it's stressful to maintain that higher dominance in that society. And I also wanted to mention briefly, just because I think it's really exciting, that hair cortisol has been studied in ancient contexts as well, as DNA methylation has as well. And I can talk about that later if you have questions about ancient DNA methylation. But in Peruvian mummies, they've actually been able to get cortisol that's been stably preserved in hair over millennia. And they were able to see increases in hair cortisol close to the time of death, which indicates like disease, illness, or stress related to something that led to death. And even related to relocation of people, where they measured stable isotopes, they measured cortisol and saw correlations about people who had moved into new contexts also had higher stress. That's pretty fascinating. I don't know too much detail about it, but I point you to that paper by Emily Webb in Toronto, I believe, who's doing that research. Okay, so I got excited about hair cortisol as a way of a biomarker of stress in this community. And I'll show you those results next. But first, I just want to show you quickly the results from phase 1, where we just were measuring stress exposures. So we did a bunch of focus groups in an immigrant serving organization in Nashville. This is the building where we did it. It was actually a beautiful place that had healthcare facility and legal facilities and all different kinds of benefits for immigrant populations, not just Hispanic immigrants. And we conducted focus groups with 32 Mexican-born immigrant women, asking them, what does estrés mean to you? What other words do you use for stress? I wanted to know if that word even meant anything to them in their context, and actually it did. They understood it. A lot of them said their children taught them what it meant, and it didn't mean the same thing. They didn't talk about it in the same way in Mexico, but they still had the concept. We asked what makes life more difficult, what makes life easier for them here as an immigrant versus in Mexico, and for what makes life more difficult or easier for their children. Lots of open-ended questions. Yeah.
Do you know the legal immigration status?
Yes, actually, a lot of the women openly discussed it in the focus groups, and then in the second phase of the study, we asked it directly, and they all answered the question. So in the survey phase of the question, where we interviewed them for many hours, 80% of them were undocumented immigrants. it amazed me how open they were and honest. And part of that is because we were collaborating with some of the people who work at this organization that had been leading parenting groups to help the mothers with parenting skills, actually connected us to the community, and they trusted us through that connection, and we were really fortunate to build that trust pretty quickly. Yeah. Okay, so the results of the first phase have been published, and this was just summarizing the main themes that we found in the focus groups about what stress exposures they were experiencing and what was important to them. And I summarize them here. Work-family trade-off was a big theme that came up. They argued they had less time with their families here than they did in Mexico because they had to work harder, longer hours to maintain their, to pay their rents. Their children ended up spending more time alone than they did in Mexico. They had limited freedom and mobility. They weren't able to drive as much as they wanted or move around as much. A lot of that due to fear of being pulled over by police and minimizing the risk by staying at home more. And this is before the election, so you can imagine it would be worse today. And actually that came up in the first few interviews I attended. It was worse. They said that they no longer allow their children to walk to school when they used to because they're afraid to let them walk down the street. So this is a nice quote showing one of the most common fears was that their children were spending too much time enclosed in the home. Encerrado was the word that kept coming up. They didn't get out to play as much as they'd hoped because of these fears. Or just restrictions on their time to spend taking their kids out. So they imagined that would be stressful for the children. They talked about loss of social networks, moving here versus in Mexico. They feel more lonely, they feel more alone. They had less childcare help than they did in Mexico, more isolation. And I asked them about how these fears get transmitted to their children. Do they talk about it openly with their children? And they said that even if they don't want to, their children pick up on it, and some of them do openly talk about their anxieties with their children and express their frustrations on or with in front of their children. But I also wanted to focus on positive. I mentioned resilience in my title. We had talked about some coping mechanisms and some resilience factors that came out. They talked about creating new social networks here that didn't exist in Mexico. So they didn't exist when they first got here. So they make new family with the friends through their churches and communities. They sought support through religion. They sought help from community resources in government assistance, community organizations. The parenting group that I mentioned helped them navigate school systems. So the main conclusion from that first phase was that their caregiving of their children was really shaped by all these larger social forces that limited the time that they had to spend with their children due to long working hours and low pay, and different forms of institutional racism that actually affect what jobs they could get, how much pay they could get, and that trickled down to affect how much time they could care for their children. Their coping mechanisms were really important in buffering these stress exposures and could be fostered through interventions. And I just think maternal immigrant stress is an understudied social determinant of health that needs more attention. Okay, so I mentioned a few possible interventions that could come from this research, more counseling programs, group social supports that could increase social support, educational programs. A lot of the mothers were seeking education but couldn't attain it because their children were without childcare. Macro level changes to policies could help the situation, things that would give them more time, maternity care, better working conditions, better pay. I didn't want to focus too much time on this. I wanted to show you quickly the cortisol results so we have time for questions. So I want to show you quickly the cortisol results now. This is the sample that we had. It's not a big sample, so it does have the problem of low power, but it is an exploratory sample that gives us some hypotheses that we can look at in larger studies. So the total sample was 81 families that we did these extensive interviews and biological collection on. The amount that actually had hair long enough to donate and the hair successfully assayed was only 57 children. And in the first phase, we only looked at cortisol in the children. We're going to do it in the moms and the children in the next phase. But we found most of the children were born in the US, 72%, and most of the mothers were born in Mexico, though a lot of them were from other Central American countries as well. And 83% of the mothers were undocumented. So a vast majority were undocumented. And then There wasn't a great deal of diversity in social economic status. Most of them were very poor, less than a high school degree, 70% of them. It was similar in the cortisol subsample. I asked about subjective SES, where they felt they were positioned in a subjective ladder in society relative to other people, and most of them had a pretty low subjective SES, 4 out of 10. And I even tried asking this with children, which I didn't think had been done much before, and I simplified it for kids under age 8, asking if they felt poorer, the same, or richer than other friends and family they knew, and most of the kids just said they felt the same. I'm not sure the question worked that great with children that were so young, but it was interesting to see. Okay, so here's the few hair cortisol results that I think are pretty exciting, and these are not yet published. We saw below high school versus high school degree in the moms. There was a small but significant decrease in hair cortisol. These are bean plots. They show you kind of the distribution of the number of samples that you have. So I like them because they are a little more informative than a box plot. You can see here, slightly lower cortisol in the children whose mothers had a slightly higher cortisol in the children whose mothers had less than a high school degree, and slightly higher cortisol in the children whose mothers rented their home versus the mothers who owned their home. We also saw a correlation with acculturation factors, like the preference they had for English over Spanish and the years they had lived in the US. So a slight decrease in hair cortisol with the children whose mothers had a preference for English over Spanish, and slight decreases in cortisol with the duration of time the mothers lived in the US. I also saw some interesting correlations with stress factors. So we asked hundreds of questions about stress. This is one example of a set of questions about stress they had over about their children's health status, access to health care for their children, dental care, mental care for their children, in a cumulative score. And once you get to a certain level of stress about your children's health, the cortisol increased. And then among the documented status, there wasn't a significant difference. There were a lot more children who were undocumented than documented, or mothers who were undocumented, but the children's cortisol just had a wider range of variation in those children. Yeah.
Why are there so many high levels?
Where? Here? There's very few samples here. It's like 1, 2, 3, 4, 5 maybe. So it might just be a byproduct of small sample size that's making it look that way. Yeah, and a few outliers. Yeah, but I'd be very interested to hear your guys' thoughts about these, because I'm still at the beginning of analyzing this, the different ways of analyzing it and thinking about it. I know some people interpret lower cortisol to be a blunting effect of stress response, and others think of higher cortisol as higher stress. With hair cortisol, I think it's more, you can't really capture that blunting effect because you're not getting a diurnal rhythm, you're getting a chronic exposure. So I'm interpreting higher cortisol here to mean higher stress. I don't think we can capture the blunting effect you get with the saliva. Okay, the last one I was most excited about was a positive psychological attribute of optimism actually being highly correlated with the child's hair cortisol. So if the mother was more optimistic, the child's hair cortisol was lower. And I think that's really fascinating. I'm going to look at the social support measures I have as well. I haven't had a chance to do that yet. And we are measuring these things again in our third phase to see if they've changed and if cortisol levels have changed with them. So just a brief preliminary conclusion of these results is that the mother's SES and some measures of it, not the income necessarily, but whether they own their home and whether they had an education, affected or was associated with the cortisol in the children, and optimism was associated with the cortisol in the children. So if these could be replicated in a bigger sample, I think that they suggest that stress and coping factors really are putting children at risk and or protecting children from the effects of the maternal stress on their stress response systems, which could have lots of downstream consequences for mental health and cardiometabolic health as well. Okay, so the final phase, I just wanted to mention, we've gone back to Nashville, we've started collecting more data, a lot of the same questions and a bunch of new questions too. Because now the children are entering puberty, so I figured we better make sure we know what stage of puberty they're at, which could affect methylation and cortisol as well. We also are getting more measures of discrimination, more measures of ethnic identity, which have not been well studied in Hispanic groups, how well they affiliate with their ethnic identity, how much it matters to them, how they think others view their group, which I've seen some interesting talks about affect the way they perceive discrimination when they experience it, based on their worldview of whether others are racist towards their group or not. Youth optimism scale that we've added, and lots of questions about politics, just like, do they talk about it at home with their parents? Do they talk about it in school with their teachers? Are they aware of these political changes and how it might affect their families? It's a picture of my grad student measuring one of the children's height with a stadiometer. And this is the final slide. I just wanted to show you, I picked out a few interesting quotes from the first 20 interviews we did in the follow-up phase of just this one question of what's your biggest worry on an average day? And it'll be interesting to compare this to the first phase, because a lot of kids talked about their parents getting arrested, parents having to leave, gun violence, which surprised me, or getting separated from their family. And then some kids' biggest worry was just about soccer practice and getting good grades, which we saw a lot of in the first phase. And I'll be curious to see if that trend moves more this way in the second phase, or if there's still kids whose grades are their biggest worry, which makes me feel good. But I have a feeling we're going to see more of this. Okay, so thank you, and I'm happy to take any questions. Yes.
Very cool talk.
Thank you.
You mentioned that a lot of the hair, for some reason, it wasn't able to be assayed.
Yeah.
What sorts of things made it?
There were only a couple, like 2 or 3 samples that didn't work well, and part of it is if you don't get enough. So I was new to HairCortisol. We wanted to be careful how much we took. You have to cut it from the scalp. In the posterior vertex of the head, right in the back underneath, is the most consistent way to collect it. And we didn't always get enough, especially with boys where it's very short and hard to cut. Also, we taped it with Scotch tape, which I later learned is not good because Scotch tape is sticky and the residue is hard to remove. Better to use painter's tape, those kinds of things. And then not everyone wanted to give hair, and not everyone had enough hair to give to. Yeah. Other questions?
The social support measures that you collected, can you just tell us a little bit about them? Is it institutional support or kid support, and if the latter, how did you measure it?
Yeah, it was mainly questions for the mom. Although in the first phase, we did have a lot of questions about the kids' friendships. How many close friends do you have? I tested out a social network set of questions, asking them to name all of their 10 closest friends, the moms and the kids, and whether they had their friends over to help them with homework, whether their friends spoke English or Spanish. So different questions for the kids and the moms. For the moms, we asked a lot about, do you get material support? Could someone help you with money if you needed it, with childcare if you needed it? So a lot of them were my own questions that I designed for the community, and then we had some standardized psychological scales of like material support. I forget the terms.
Did you ask about kid support?
Do you mean childcare?
No, kin. Their
kin.
relatives. Yeah.
Well, we asked who their top 10 people they go to for different kinds of support, and sometimes they would tell us if it was siblings or family, and we asked questions about family being close by or not. So some of our questions address that, yeah. And I have yet to analyze the social support data really to say what mattered and what was measured very well. Yeah. Yes?
You talked about obesity a lot previously, but I was wondering if in this study if there was any link between the hair cortisol and like BMI.
That's a great question, and it has been linked in prior studies. In ours, not so much. And I'm not sure yet why that is. It could be problems with the measurements. or it could just be that it's not consistently correlated with hair cortisol. But I didn't see a link. Yeah. I looked at it with height and weight, BMI percentile, BMI Z-score, and I didn't see an association.
Do you think you might be able to do a comparison since you're gonna have like earlier data
Yes,
and late
yes.
data?
Yes, I definitely want to compare cortisol across time, methylation across time, in the same. We're looking in candidate genes. We're also looking. We're going to look genome-wide and at BMI. I only measured blood pressure in children above age 11 in the first phase because a pediatrician told me that was the most stable period to measure blood pressure. But then I regretted that we didn't try to measure it younger, so this time we measured it in everyone, and we'll have more. So there were only 17 kids where we had blood pressure measures in the first phase, and it didn't associate with anything. But this time I hope we'll have more, and we might see something.
Very cool.
Thank you. Yes?
So I have a kind of a relatively simple interpretation of results
Uh-huh.
question, and then a more broad conceptual one. First, so I can certainly see how a mother's forecast of her child's future could shape the mother-child interactions and the home environment, and could be reflective of a variety of exogenous circumstances that the mother is experiencing. So, you know, it makes sense to say, well, the child's hair cortisol level is a reflection of either mother's interaction with child or mother's assessment of larger circumstance, something like that.
Mm-hmm.
But it could also be the other way around, right? That is, kids who are doing well have mothers who, seeing that their children are doing well, are optimistic about their kids' future, right? So, yeah, you know, my child is, you know, healthy and getting good grades, and I mean, most he's worried about is, you know, whether he's going to get a B or an A, or, you know, get hurt playing soccer or whatever, you know, I mean, these kinds of things, right? So I'm optimistic about my child's future because I'm kind of watching my child's progress over time, right? So,
Yeah, I think that's an interesting way to examine the data that I'd like to do. Look at the children who are successful despite difficult circumstances as a way of looking at resiliency and seeing how that affects the children's health. But I like your idea of the reverse causation. I hadn't thought about the successful children influencing the mother's perspective.
I mean, if you look at, and you're probably more familiar with this literature than I am, but if you look at parents who have children who have significant handicaps, right, and where it's clear the child is going to be facing challenges throughout life, parents of, you know, children with severe autism or something like this, what they talk about a lot is, you know, what's going to happen to my kid when I die, right? Who's going to take care of my child, right? So they're very pessimistic about their child's futures. It's not because they're creating that future for their child, they're just assessing what the future is. Right? So it could work that way.
Yeah.
Conceptually, and you sort of, you showed your cards a little bit on your perspective on this at the very beginning in framing the presentation, but you didn't sort of lay out the whole logic, which is, you know, where the adaptive mechanism is or isn't in this whole system that you're studying, right? So it seems to me there are a number of possibilities, and I'm just curious, since you're the expert on this literature, where you see, which you see as the most likely, right? So one possibility is that the whole methylation system isn't adaptive now and never was, right? That this is, for some unknown reason, it's some consequence of something else.
Dysregulated consequence of overload of stress.
Yeah. I mean, it's a little hard for me to envision why you have a complex molecular mechanism that never did anything. So another possibility.
Well, it does have other roles, right? I mean, it controls how cells differentiate, and
sure, but why it is responsive to stress in that regard? I mean, you have to have some constraint on optimality argument that says that the impact of stress is just a byproduct or a misfiring or a constraint on how the whole system operates one way or another. And it's possible that that's the case, but I can't envision it. And so if you can help me to see that perspective, I'd be interested. And another possibility would be that, you know, in the environments in which this system evolved, it was adaptive up to a point, and then maybe got dysregulated occasionally in past environments, but it's getting dysregulated a lot more because there's a phylogenetic mismatch here, right? Where the world is really, really different than the world in which the system evolved. I mean, that sounds like a plausible possibility also, but I'd be curious to know what you think is different, right? I mean, how is it, it's not that our ancestors had it easy, right? And it's not that they weren't subject to, you know, I mean, worrying about ICE is I'm not trivializing that at all, right? I mean, obviously that's discrimination and racism are very real and impact people. But our ancestors were also, you know, subject to rating by outgroups, right? I mean, bad stuff has been happening to people as long as they've been people. So the question is, if you think it's phylogenetic mismatch, what is it that you think has changed to cause the system not to be adaptive?
I'm not convinced it is mismatch.
Okay.
But if it were, I think the biggest change is diet.
Okay.
Access to too much calories. And I'm not talking about diet really much here. I'm talking about stress. But even still, I think the mismatch could be that children that are under stress have more reward-seeking behaviors because they're trying to gratify more quickly in short periods of time, and then they're getting a glut of calories that they might not have had access to in the past, and those reward-seeking behaviors could lead to tendencies towards obesity and diabetes. So in that sense, I think there could be a mismatch with the past because we have so much easy access to calories. But I'm not sure that mismatch is the story I would use to frame the stress response. I think, yes, you're right. It's a complex genetic mechanism, epigenetic mechanism, that has locus-specific changes that are passed on, at least from mother to child in at least one generation. I argue, I don't know that we have evidence that it's passed on through the germline across multiple generations, but at least the mother's behavior and the mother's environment in pregnancy and early childhood, I think, affect the methylation of the offspring in a way that passes on some kind of signal to the offspring in a very locus-specific, timing-specific, complex way that I agree with you is not totally random and not just a byproduct. I think it could have a role of passing on a signal of the mother's environment, but we have yet to find what that exact role might be. The only hint of it that I have is this increased vigilance, increased sensitivity, right, to threats. And there could be a mismatch somewhere in there where we're so sensitive to threats, more so than we need to be, that it leads us to develop mood disorders and anxiety disorders that aren't beneficial. But maybe in the past, the sensitivity to threat was more important, and it could have helped us avoid problems with strangers and predators, who knows. It's complicated. I haven't thought it through all the way yet in terms of this project. I think there will be a role for this adaptive mechanism in the story. And maybe there is a little bit of a mismatch in diet, in stress response, being overly sensitive, where that could have been a benefit in the past. I'm still thinking it through, and I'd be happy for your thoughts. Yeah, if anyone else has thoughts about it. I frame this work as dysregulation because I think it's overload of toxic exposures that are causing harm to children. So I don't think there's a benefit to this in these children. If there were, I don't know how we would detect it. Yeah.
Just to follow up briefly on that, but you're measuring it in terms of health outcomes, not in terms of fitness, right? So, you know, and given the many differences, including family planning between the contemporary environment and the past one, it's certainly possible that that even if you were to measure fitness, you wouldn't necessarily see anything at all. But one theoretical possibility is that you know, tracking people onto a faster life history trajectory with greater vigilance paid fitness dividends in small-scale ancestral societies at the expense of longevity.
Yes.
Right?
I just think that's so much easier to test in an animal model where you can get the whole life course more quickly and across generations. It's so hard to know if that's really happening in humans.
Sure.
But yeah, it's definitely an interesting theory. And I don't think we have evidence either way yet. Yeah.
Just, this is more of a comment than a question, but
Yes.
just following up on Dan's thing, it seems to me that, you know, I mean, often adaptationists would think about these kinds of things in sort of stress responses, plastic stress responses, or adaptive for whatever reason. But it does seem to me like that there's a, if you flip it and think processes of tissue differentiation and things like that are actually difficult to achieve optimally, so that the normal functioning of the system is attempted to be optimized by natural selection or something like that. There's just a lot of ways to mess it up through stress and things like that. That seems like a very plausible hypothesis for why most of this could be not.
So the stress isn't adaptive, it's a way of messing up the normal regulation of
Which
these.
seems
Yeah.
you're kind of
That's a nice way of thinking of it.
that seems like that could easily be an explanation for this stuff that might be very difficult to tease apart from adaptationist hypothesis, because they might have similar,
Yeah.
outcomes or whatever.
Yeah, the only argument against that it's messing it up, is that we do see some consistency in the locus-specific patterns. So there's a complexity there that doesn't seem random entirely.
Yeah.
But I think we need more evidence to show whether the changes across children are random or not. Yeah, that would be a good way of getting at it.
But I think even that is to I think you can salvage Clark's deflationary account in light of that observation by saying, well, ways of messing it up is a loose description for, in order to have the proper gene expression for tissue differentiation, right, you have vulnerabilities in the system that will be patterned in the way that they respond to insult, right? So you'll see specific patterning under adverse circumstance. It is not an adaptive response, it's just the way that the system is designed. So, analogy might be something like, well, you have a particular structure in bone, and that structure causes pattern fractures when the limits of the bone strength are exceeded. And the patterns will be fairly consistent over time. That consistency isn't because there's anything adapted in the fracturing, it's because that's the way the bone is patterned.
A byproduct of the pattern of the bone.
Yeah, I mean, it could be. I'm skeptical of that account. But
I find it very hard to tease apart
Yeah.
these hypotheses. Yeah. Other questions?
Hi, so have you measured in this, or are you thinking about measuring A1C?
Glucose, or you mean for risk of diabetes?
Yeah, I mean, there's also some stuff relating it to certain behavioral measures. Maybe we can talk more about that later.
Oh yeah, I'm not familiar with that. I'm not measuring that in part because I don't have blood.
Right.
I don't know, maybe you can tell me if you can measure it in anything else, but I don't think that you can. We do have saliva, which has blood cells. So saliva is a mix of blood and epithelial cells, so it's in between. But I don't know if there's an assay optimized to measure that in saliva, probably not.
Okay, and then another thing, just curious. Okay,
go ahead.
how about locus of control measures? Have you thought about that? I'm not sure.
Is that a psychological term? Can you explain more?
Oh, more about so whether or not you feel like you have control over your own life is more external, internal. Do things happen to you, or do you make things happen?
Yeah, I don't actually have measures of that, but that's a good area. There's so many things to measure, and interview fatigue was a big concern of mine, because we ended up, when it went over 2 hours, we often opted to let the mother continue the interview on the phone at a later date and just get all the biological measurements out of the way, because we just couldn't even ask all the questions we had in that time period. But yeah, we left that out.
Yeah, no, the reason I mentioned is the stuff you have on optimism is really interesting. There's a literature on locus of control and other psychopathology.
Might be related to the optimism.
Yeah, that's what the connection is.
Yeah, optimism I find interesting. A lot of people are critical that it's not really a measure of resilience because it's a personality trait that's pretty stable. And if you want to think of resilience as a process that can change, or that's a way of garnering resources, optimism's not a good measure of that. But I think it's an interesting measure that can actually change over time and is patterned by early adversity. So in another study I'm doing, we found optimism to be patterned by early adversity and predictive of health behaviors. So I think it's fascinating. It's hard to know which direction it's going, whether their life experiences are influencing their optimism or the optimism is influencing the health of their children and their behaviors. But it's something important in these complex pathways that we can actually intervene. There have been interventions that could actually increase optimism by having people write about their futures or think about their futures in certain ways. So I don't think of it as a static trait necessarily. I think of it as something that can be changed over time. And in that way, I like the idea of it. Other questions? I'm curious, just one question I have for you guys. When I presented the article by Marzi et al. About lack of any significant findings genome-wide in the biggest, most comprehensive study to date, were you guys surprised, disappointed? Do you think there's problems with the study, or do you think it's a signal that we should be moving in a different direction?
I mean, I found your argument about blood is the tissue of interest a compelling one. Right?
Yeah.
It's kind of looking under the lamppost, right?
Yeah. We look where we have samples.
Right, exactly.
Yeah. Yes?
I also find that GWAS are not the most compelling data. I don't know, correlation is.
Yeah, you're never going to get causation
Right.
with that, with observational studies.
Yeah.
That's true. Did you have a?
Well, I was similarly, I was going to say, isn't that actually a fairly typical finding for GWAS studies? That there's some small or no effects for.
Yeah. The other thing
So
is,
it doesn't
even when
make-
we do find small effects, they're not consistent across studies. And yeah, same thing
Which
with
just makes
GWAS
it
and
seem like there's nothing
EWAS.
particularly special about this. I mean, you shouldn't be any more or less
Disappointed.
skeptical or disappointed about this particular domain than anything else that's studied.
Yeah, I think it's even harder in social behavioral questions because it's hard to measure those so accurately than with more diet or other kinds of toxins. But you're right, it's similar to GWAS findings, yeah. And maybe we just need more nuanced measures, maybe we need different tissues, maybe we need more experiments with animal models to get at causation. It's not the only way to go, for sure. Okay.
All right, we are exactly.
Thank you.